rolling.
This lecture is about energy. We eat food, for example, sugar. How do you suppose we get the energy from sugar? Do we burn it in a stove? Or is there another way? Well, sugar, for example, glucose, the simple sugar, has a lot of energy. If you completely oxidize glucose, you get 686 kilocalories per mole of energy. That is, delta G is very large and negative. When you take sugar and oxidize it with oxygen completely to carbon dioxide and water. So one way to completely oxidize glucose is to burn it. But that's not how cells work. The problem was, how do you harvest the energy efficiently? If you burn it, it would all go as heat. So what the cell does is it takes the energy in small steps. So little reactions sum up to be one big reaction. If you wanted to burn sugar, you'd have to provide a large activation energy. So if free energy is on the y-axis here, and the reaction direction is on the x-axis, the activation energy is shown as this hump here. That would be the match, the kindling. And all the energy would be released as heat. The cell does a controlled, stepwise oxidation of sugars, so they efficiently harvest energy in multiple small steps. How do they do that? Cells are smart. They have activated energy carrier molecules. So there's several of these, and ATP is one you've heard of before, and ADH is also one you've heard of before. We're going to use those to get energy from glucose. So what I hope you understand today is the forces of energy. What thermodynamic forces are involved in getting that energy from sugar? I'd like you to know the difference between energy storage molecules. That would be fats or starches or sugars. Those store the energy. And energy carrier molecules. Those carry the energy from one place in the cell for one reaction or another. And I'd like you to understand the flow of energy from catabolism, that is breaking stuff down, or oxidation, into energy carrier molecules. Finally, reaction coupling is used. And that is, you use the energy from one reaction to make an energy carrier molecule so that you can use that energy carrier molecule later. So you harvest the energy through coupled reactions. The Earth is very, very old. The Earth is very, very old. 15 billion years ago, there was the Big Bang, and 4.6 or more billion years ago, the Earth was formed. Then there was the RNA world, and out of that came the first living cells, about 3.6 billion years ago. Some of those learned how to do photosynthesis, which I'm going to tell you about when we talk about chloroplasts. Photosynthesis released oxygen, which was precipitated by ferric ions in the ocean for 2 billion years. And then all the iron in the ocean used up the oxygen in the ocean. All the oxygen reacted to the iron in the ocean and oxygen started building up in the atmosphere. That's why we have oxygen in the atmosphere, and it's just because of the sun and photosynthesis. Because we have oxygen in the atmosphere, we can use that oxygen to oxidize the energy storage molecules, fats and sugars, and gain energy. Oxygen is a really good electron acceptor. Oxygen loves electrons. So the oxygen released from photosynthesis is used in respiration for catabolism, for breaking stuff down, for combining with sugars to make water and carbon dioxide. So we have energy catabolism, breaking stuff down, and anabolism, making stuff. So plants make sugars from carbon dioxide. We use those sugars to gain energy so we can walk around and study and give lectures. So the anabolic pathways are the ones derived from the sun. Those are carbon fixation, making things, synthesis, synthetic pathways. The catabolic pathways are what I'm going to talk about today and in the lecture about mitochondria. That's breaking down things. And there are two types. What I'll be focusing on is the aerobic, or in the presence of oxygen. You can also have anaerobic catabolism in the absence of oxygen. And there are some bacteria that are really good at this. Yeast can do that too. So plants make food in the form of sugars, carbohydrates. Carbohydrates are sugars and starches, anything with carbon, hydrogen, and oxygen. That's a carbohydrate. And fats. Fats have long chain fatty acids, and I've talked about those in the membrane lecture. So starches are branched glucoses, which are linked as polymers, and so is glycogen, which is stored in your liver. So glucose can be snipped off one at a time and then used by the mitochondria and by cells. Fats are made also in plants, and those are useful for getting a lot of energy, but they also require oxygen. So the whole energy scheme relies on oxygen, and so it's called cellular oxidation. So what is oxidation? You remember oxidation from your freshman chemistry. Oxidation is loss of electrons, OIL. Oxidation is loss. And reduction is gain of electrons. So if you have an oxidation and reduction reaction, it's always a redox pair. It's called a redox reaction, oxidation and reduction. So let's just review what oxygen does during oxidation. Oxygen is not the only thing that can oxidize things, but oxygen is greedy for electrons, and so it's a good example. It produces energy, and that means that the delta G for the reaction is negative, so it will go forward. Let's examine burning methane. That is, methane combines with oxygen to form carbon dioxide and water. The oxidation state of oxygen in its elemental form is 0 by definition, and the oxidation state of carbon here is -4. So it's got 4 extra electrons that oxygen is going to take because oxygen is electronegative. Oxygen is hungry for electrons. So the oxidation state here of oxygen in CO2, or carbon dioxide, is -2. So oxygen gains the electrons, whereas carbon, or methane here, has lost those electrons. So methane is oxidized, it has lost its electrons, and oxygen is reduced, it has gained its electrons. So here it's shown that carbon loses its electrons. These 8 electrons go into oxygen, and oxygen is then reduced. So that's a little freshman chemistry to remind you. We're not going to be burning methane in cells. What we use is sugars and fats. Just to remind you, the energy currency, that is the molecules that carry these electrons, these high energy electrons, are NAD, nicotinamide adenine dinucleotide, NAD, and NADPH. We'll see NADPH later. It just has a phosphate group. That's used for anabolism. NAD or NAD plus or NADH are used for catabolism by mitochondria. Also, this one, flavin adenine dinucleotide, or FAD for short. This guy is also an energy carrier molecule. So if we focus in on what gets oxidized and reduced, the reduced form of NADH or NADPH is this bond right here. This hydrogen adds to this double bonded ring, and that's the reduced form. So that's carrying a proton and 2 high energy electrons. For FAD, this structure here carries 2 protons and also 4, count them, high energy electrons. So this is called FADH2. That's the reduced form, carrying those high energy electrons. So when we're oxidizing glucose to carbon dioxide and water, we're making reduced NADH and reduced FADH2 as intermediate steps to harvest the energy from the oxidation of glucose. And finally, at the end of many small steps in mitochondria, oxygen is the final electron acceptor. So just like burning methane, oxygen is hungry for electrons and will accept the electrons. So we have a series of small steps mediated by enzymes. Enzymes will catalyze these small steps and couple the reactions to these reduced electron carriers and synthesis of ATP. And the final electron acceptor is oxygen. So we'll focus on ATP, our electron carriers, and then finally acetyl-CoA. Acetyl-CoA is the thing that carries the acetyl group. So you've seen these before. ATP is the universal energy coupler. The delta G for the hydrolysis of that terminal phosphate is highly negative, and it's made even more negative in cells because the concentration of ATP in cells is quite high, thanks to mitochondria. And that's because of the thermodynamic equation shown here. And you should review question 3.8 in your book so you can understand why the delta G is even more negative in cells because the concentration of ATP is high. Acetyl-CoA accepts and transfers these small acetyl groups. Those are 2 carbon units, and there's a high energy bond here which can be used, the energy of the bond can be used to make stuff, to make carbon compounds. So they always come in pairs, the carbon compounds. That's because of acetyl-CoA. It's a nucleotide. It's got an adenine, a ribose, phosphate, and then this structure here. The important part is that the acetyl groups are transferred. So we've just had a big meal. I hope you had breakfast. Breakfast is the most important meal of the day. And you might have had roast beef and maybe not. But we're going to digest that into proteins, sugars, and fats. And the part that I'm going to be talking about today is the first steps which occur in the cytoplasm of the cell. So this whole round thing is the cell, and this little mini round thing is the mitochondria. In another lecture, I'll talk about what happens inside the mitochondria, but for today we're going to focus on taking up glucose and converting it to pyruvate, harvesting some energy along the way. That's called glycolysis. That is lysis of glucose. So glycolysis is a series of reactions that excited biochemists all through the first part and the middle part of the 20th century. Let's get the century right. So energy has to be invested in the form of ATP in the first few steps. But then it's gained back later because these steps are used in coupled reactions to release energy in the form of ATP and NADH. So you have to put in 2 ATPs to get out 4 ATPs and 2 NADHs and then 2 pyruvates, which will go on later to give you a lot more ATP. So it's a series of steps. All of these are known, and some of these are controlled in various ways by enzymes. So I'm not going to go through every single one of these steps, but I'm going to focus in on just one step here. That is step 6 and 7, because it illustrates the principle of reaction coupling. Reaction coupling. So what this step does is it converts an aldehyde to a carboxylic acid. Okay? So this is an oxidation. An oxygen is reduced, it's added to this carbon. And from this you get 1 molecule of NADH and 1 molecule of ATP. It's a high energy reaction. So what is reaction coupling? This occurs in a couple of different steps. The first step, the carbon-hydrogen bond is broken, and that energy, that's this arrow here, the energy that's released by this carbon-hydrogen bond being broken, is coupled to forming this high-energy phosphate bond. Okay? So an intermediate is formed in which the phosphate is attached to this carboxyl group. In the second step, this energy is released, that's this brown arrow, and the phosphate is transferred to ADP to form ATP. So in the first step, the reaction of forming this high energy bond is coupled to the hydrolysis of this carbon-hydrogen, and at the same time, NAD is reduced to NADH. So you've got two energy storage molecules here. One is our carrier molecule, the other is this high energy phosphate bond, and then this phosphate can be transferred to ADP to form ATP. So that's energy coupling. You form high energy intermediates to gain all of this energy and transfer it to something else for use later. You can read in your book about other of these reactions and how reaction coupling is used to harvest energy. The end result of glycolysis is the formation of pyruvate. That's this guy here, a simple 3-carbon structure. Pyruvate's very useful in this cell. You can ferment it. You can ferment it in yeast to make ethanol, in muscle to make lactate. fermentation requires enzymes, and it allows regeneration of NAD+. So it's useful when you go anaerobic. In contrast, you can oxidize it, which we'll do in the TCA cycle later. So fermentation without oxygen allows regeneration of this carrier molecule, so it can be used in glycolysis. In muscle, it forms lactate. So you get ATP, you form pyruvate from glycolysis, and you form NADH. And pretty soon you run out of NAD, so fermentation allows you to form this intermediate lactate and regenerate NAD so that glycolysis can continue, and you can continue to make ATP. Lactate, you might have heard if you exercise a lot, you build up lactate if you go anaerobic, and it makes your muscles sore if you do this. So this is when you do a sprint. When you sprint, you form lactate. In yeast, they don't have muscles, but they use the same principle. They use glycolysis, and they make ATP, and they also use up their NAD+. So all the NADH needs to be fermented to regenerate the NAD+, and they make this stuff called ethanol. And ethanol is useful for a variety of things in man. Lactate and ethanol are the products of fermentation. Pyruvate can also be made into glucose in the liver, and that's called gluconeogenesis, the making of new glucose. And that's essentially glycolysis going backwards. So glucose to pyruvate is going down in this diagram, and all these intermediate steps generate energy. Pyruvate can be made into glucose. It requires energy. So if you have excess energy after a meal, or if you're digesting your muscle during a fast, you can make glucose because your brain prefers glucose as food. So gluconeogenesis requires pyruvate, NADH, our high energy electron carriers, ATP, GTP, and water. And it generates glucose and the energy carriers depleted of their high energy bonds or electrons. There are 3 steps in glycolysis and gluconeogenesis that are highly exergonic. In other words, they have a large negative delta G. These steps are bypassed in gluconeogenesis with different enzymes, and they're also control points or regulatory steps in the 2 pathways. When you study biochemistry, you'll study these further. So let's now turn to pyruvate and making more ATP. So we're going to go into the mitochondria now. I've taken you from sugars through glycolysis, glucose to pyruvate, and that's all in the cytoplasm, the cytosol of the cell. Pyruvate is taken up into mitochondria and turned into acetyl-CoA, and through the citric acid cycle, or the TCA cycle, generates a lot more ATP, and later it also generates these NADHs, and later these NADHs are used to make even more ATP. Pyruvate is oxidized to acetyl-CoA. It releases 1 carbon. So you go from a 3-carbon unit to a 2-carbon unit attached to acetyl-CoA. These 2-carbon unit building blocks will be used for a series of reactions later. So 1 carbon is lost in carbon dioxide, and that's exhaled in people. you also gain a high energy electron carrier NADH. The enzyme that does this is a large machine with several enzymes together in a kind of particle. It's a kind of protein machine, and it's called pyruvate dehydrogenase. The acetyl group is used in the TCA cycle to generate energy, and that's the central pathway for oxidation of fuels in mitochondria. Again, when you study the TCA cycle, the citric acid cycle, or the Krebs cycle, those are all names for the same thing. You'll learn all the intermediates in gruesome detail. For now, I'll just tell you what goes in acetyl-CoA and what comes out. We get NADH, GTP, FADH2, and NADH. These high energy electron carriers will be used later to make more ATP. So these 2 carbon units are made into citrate, which is a 6 carbon unit, and then it gets broken down gradually and added. You go from 6 to 5, releasing carbon dioxide, and then you go to 4, and 2 more are added, and it's really a cycle. And there's some interesting problems in your book about how people figured this out. In fact, we know exactly where the 2 carbons go in the whole reaction. So this diagram traces the 2 carbons coming in from acetyl-CoA. They're added to oxaloacetate. These red carbons are the new carbons. Then these are, and then this guy is broken off, so you're snipping off, this guy is broken off, so you harvest the energy from one, releasing carbon dioxide. Then the red carbons remain through these reactions, and they become part of the backbone of the next cycle. So the whole thing moves iteratively, snipping off stuff from the side or from the bottom, releasing energy the whole way. So the important things for you to remember at this stage is that you get energy in the form of GTP, in the form of NADH and FADH2, and 2 carbon dioxides are released. So this goes around twice for every glucose molecule. Glucose is 6 carbons. You lose 2 CO2s. You end up with a 4 carbon molecule, and it goes around once more for every glucose molecule. So that's the TCA cycle. So 1 molecule of glucose, that's a simple corn syrup sugar, gives you 2 ATPs in glycolysis. You have to put in 2 and you get 4 back. You get 2 NADHs in glycolysis, 2 NADHs in the TCA cycle. And then the pyruvates are converted into acetyl-CoAs, which go into the citric acid cycle to make GTP, 6 more NADHs, and 2 FADH2s. So you get a lot of energy, a lot more than you would if you took a match to it. From fats, you even get more energy. That's why fats are so good. Fats have these long hydrocarbon tails. Long hydrocarbon tails are oxidized stepwise to take 2 units of carbon off at a time and transfer them into, guess what, acetyl-CoA. And this is called fatty acid oxidation. This occurs in mitochondria. First, an acetyl-CoA is added to the long chain, and it's snipped off 2 by 2 until you're down to the shortest unit, which is 2. So that's why fatty acids and lipids The chain length is usually an even number. It's not an odd number. You don't have 17. You'll have 16, or 14, or 20 carbons per tail. So you generate FADH2 and NADPH, or NADH, and acetyl-CoA. So you don't get any ATP from this, but these are used in mitochondria later to make ATP by ATP synthase. So every 4 carbons, and fatty acids can be 16, 18, 20, and more, every 4 carbons gives you 2 NADHs, 2 FADH2s, 2 GTP, 6 NADH, and 2 FADH2 in the TCA cycle. So you really get quite a lot of energy from each fat molecule. That's why fats yield more energy, because you get all of these carriers. And next we're going to see how mitochondria use this energy to make ATP. And it's amazing, it's an amazing story. And I encourage you to look at the movie of ATP synthase. That in this diagram is shown here. We've gone through glucose to pyruvate, that's glycolysis in the cytosol. We've gone from pyruvate to acetyl-CoA through the citric acid cycle that occurs in the mitochondria. And next we're going to talk about the reduced energy carriers, NADH, FADH2, making ATP in a process called oxidative phosphorylation. That is, you take reduced molecules, oxygen is put in as the final electron acceptor because it's greedy for electrons. And you make ATP. That's for the next lecture. Thank you.